All right, welcome to Chapter 8, Patterns of Inheritance, Part 2, Mendel's Laws of Inheritance. And just a quick review, remember homologous chromosomes from last time. If you're homozygous dominant, you got both versions of the dominant allele on each separate chromosome. If you're homozygous recessive, two recessive alleles, and if you're heterozygous, a dominant and a recessive between the two. And all the alleles that are on one chromosome are said to be linked to each other, because wherever one of these chromosomes go, that's where these three different alleles are going together. The law of dominance it tells us that of the two alleles, if one of them is dominant, it means that when you have one of them in a heterozygous situation, the dominant phenotype is what you're going to see. The phenotype is the observable trait. The genotype is the combination of alleles that are there. So homozygous dominant is a genotype. Purple flowers was the phenotype. Okay, and dominant masks the recessive when you're heterozygous. So if I take, and I can show this, if I take a true breeding yellow parent with a green, with a true breeding green, so this is homozygous dominant for yellow, which is a dominant phenotype, homozygous recessive for the green, and this is the parent generation. This organism can only contribute, and they've got these backwards. This is actually over here, and this is actually here. But this organism can only contribute a recessive allele. This organism can only contribute a dominant allele, right? So 100% of the F1 generation is going to be yellow because they all are heterozygous and they all have one dominant allele, capital Y. Now, if I cross two of these, what I expect to see is that 25% of them will be homozygous dominant, 50% will be heterozygous, and 25% will be homozygous recessive, which means that 25 plus 50 that have at least one dominant Y will be yellow, and 1/4 of the offspring are predicted to be homozygous recessive and green. And right now you're saying, wait a minute, where'd you get these numbers? Where'd you get these numbers? And we're going to show these in a second. But if we talk about the ratio, the phenotypic ratio would be 3:1 yellow to green in the F2 generation. Okay, the genotypic ratio would be 1:2:1 homozygous dominant versus heterozygous versus homozygous recessive in the F2 generation. And you'll need to know those ratios later. All right. So again, if we're homozygous dominant, we've got both dominant P alleles, which is what the true breeding parent should be. If we're homozygous recessive, we've got both recessive alleles for the recessive color, which is what the true breeding white flower should be. And in the first filial generation, the F1 generation, all the offspring should be heterozygous. And since they've got one dominant P, they should be purple in color. Now, how did I know what those percentages should be? Well, because we can predict them using something called a monohybrid cross. And a monohybrid cross uses something called a Punnett square, where we take the possible gametes, possible alleles that someone in the first filial generation can contribute, and we put it in a Punnett square, and it's kind of like a multiplication table where we just bring down the various letters and multiply them. So let me show you what it looks like. Here's our Punnett square, and it's going to be a 2x2 Punnett square. We had our parent generation. This organism could only contribute a dominant allele. This could only contribute a recessive allele. So all the members of the F1 generation had one dominant, one recessive allele, had the dominant phenotype of yellow. Okay. Now, for the F2 generation, each of these organisms can contribute either a dominant or recessive allele. So we take two of them, and we put the dominant and the recessive allele from one of them here, and we put the dominant and recessive allele from the other one here, and then we just fill it in like a multiplication. Y. Capital Y would go here. Capital Y, lowercase y would go here. We always put the capital first, so capital Y, lowercase y would go here, and then lowercase y would go here. And then if we draw in the genotype or the phenotypes, yellow yellow, yellow, because they all have at least one dominant allele, green, because it has two recessive alleles. All right, so my genotypic ratio is 1 to 2 to 1, homozygous dominant to heterozygous to homozygous recessive. My phenotypic ratio is 1 to 3 to 1, yellow to green. Now, that's what I use to generate those percentages that you saw before. Because out of 100, if there were 100 offspring from this cross, I would expect 1/4 of them, or 25%, to be homozygous recessive, and I should see the green color of the seeds. I would expect 75% of them to have the dominant phenotype of the yellow seed color, which I can see. And what Mendel did was he crossed huge numbers of pea plants, so that he wasn't getting 3 to 1. He was getting something like 2,975 to 1,112. And the more offspring he had, the more he saw that it got closer to that 3 to 1 ratio. And based on his mathematical background, this is how he worked backwards to determine what had to be happening. Okay, so from this he developed two other laws. The law of segregation is the first one, and in the law of segregation it says that the genes must segregate equally into gametes. Okay, so if we start with our in early meiosis, and we double our sister chromatids, and then we put the tetrad on the mitotic spindle, and then we pull both chromosomes from one. If we separate the tetrad, and so each of our daughter cells is going to have two identical chromosomes of this one, two of this one, two of this one, two of this one. We're 2n, because we still got 4. We started with 4 chromosomes. We've still got 4, but at the end of meiosis 1, we've got 2 copies of 2 different chromosomes here and 2 copies of the other 2 chromosomes here. If we didn't cross over, and crossing over probably occur, but and also if we do it correctly, and non-disjunction doesn't occur. The law of independent assortment states that the genes don't influence each other with regard to sorting of alleles, unless they're linked on the same chromosome. But then they're not influencing each other, they're just together on the same chromosome. And what this means is we're likely to see every possible combination of alleles for every gene, and it should be equally likely to occur. And he illustrated this with a dihybrid cross, where he took 2 true breeding parents that express different traits for 2 characteristics. Okay, and so in our dihybrid cross, we're going to differ the parent varieties in 2 characters. One's going to be homozygous dominant for both traits. The other's going to be homozygous recessive for both traits. So let's stay with the P, and let's consider round versus wrinkled, because round is dominant, wrinkled is recessive. Let's consider yellow and green. Yellow's dominant, green is recessive. All right, so here's our parent generation. We've got homozygous dominant for yellow, homozygous dominant for round. We got homozygous recessive for yellow and round, which would give us a green and wrinkled. This organism is going to contribute a Y and an R, but they're identical. And this organism is going to contribute either a lowercase y or lowercase r. It doesn't matter which one, because they're identical. So in the F1 generation, all the organisms are going to have a dominant yellow and a recessive yellow, a dominant R and a recessive R. So they're all going to be round and yellow, but they're going to be heterozygous for both characteristics. And in a dihybrid cross, now we're going to cross 2 of these F1s, which is YY, big Y, big R, little R each. And we have to look at the various gametes that they can produce. Well, this organism could give us a big Y with a big R. It could give us a big Y with a little Y with a big R, and a little y with a little r. And this organism, the same thing. So this time our Punnett square is going to be 4 by 4, because big Y, big R, big Y, little r, little y, big R, and little y, little r. And then when we fill in, okay, big Y, big Y, big R, and so on as you work your way across. Now, the thing that Mendel was focusing on in this was, again, the phenotypes of the offspring in the F2 generation, because that's what he could see. All right. The only way you could get around wrinkled is if you had recessive alleles from both, homozygous recessive for both. Okay. If you have at least one dominant allele, you would expect to be round and yellow. So 1, 2, 3, 4, 5, 6, 7, 8, 9 are round and yellow. If you had at least one dominant yellow and both recessive for wrinkled, you would have 3 that were round and wrinkled. And if you had both recessives for round or both recessives for the green color and at least one dominant for round, you would have 1, 2, 3 that were round and green. And seeing this 9-3-3-1 ratio, and again, Mendel's working with thousands and thousands of offspring from the F2 generation. His numbers are more like, 9,100 versus 2,912 versus 2,813 versus, 905. They're not going to be exactly 9-3-3-1, but the more you do, the more crosses you do and the more offspring, the closer you're going to come to that 9-3-3-1 phenotypic ratio that he would expect mathematically from a cross like this. Okay, so that's how Mendel was able to put together his laws, and we found today that they still work. Now, what if we have a pea plant and we don't know its genotype? Okay, so it's got a yellow seed, but we don't know if it's dominant, homozygous dominant, or heterozygous. Well, we could cross it with a homozygous recessive, because if it's homozygous dominant, this organism can only produce a recessive allele. So this would have to be a dominant allele if all the offspring had the dominant phenotype. But if half of our offspring have the recessive phenotype, then that's an indication that this is recessive, and this organism was heterozygous because it had to have a recessive allele for these two organisms to both be homozygous recessive. So that's what we do with a test cross. So why would we want to do a test cross? Well, it might not matter with a pea plant, but it matters with certain breeding we do with animals and livestock. Let's say we've got a dog breeder, and he wants to raise black lab puppies using a black male as the sire. Maybe this dog's a really good dog. He's super friendly. Maybe it's a service dog, and he's got great characteristics of that. Maybe he's got a great nose, and they use him as a drug sniffing dog or a bomb finding dog. But for whatever reason, they want to breed this black male. But before they do, they want to make sure he's homozygous dominant for black coat color, because if he's heterozygous, he could have chocolate lab offspring. And it turns out they have a shorter lifespan, and there's a couple other issues that chocolate labs deal with that black labs don't. And so before he lets the dog sire multiple litters, he does a test cross. And he takes a chocolate female, because if she's a chocolate female, she has to be homozygous recessive for coat color. Okay? And he doesn't know what he is. He knows he's got at least one dominant allele, but she's got both recessive alleles. And in the litter that produces 8 puppies, 7 have a black coat and 1 has a chocolate coat. And so based on this, what do you think the genotype of the sire is? Realizing I can't hear you. Give you a chance to think about it. Well, the only way that one of those puppies is chocolate is if it got a recessive allele from the father, because that's all the mother can give, but the mother can't give both recessive alleles. So the father has to be heterozygous. And you're saying, well, gee, we just had a litter and we've got, 8 puppies that, we don't know that we really want to do anything with them. Well, they're going to make good pets, and you're always going to find a home for chocolate labs and black labs, regardless of their genotype, because most dog owners are going to have the dogs neutered or spayed, and they're not going to raise litters anyway. But if, sometimes if you've got a dog with really desirable characteristics, he might sire hundreds of litters over his lifetime. And so doing a test cross makes sense with animals and livestock that we might want to have a tremendous number of offspring from because they have some characteristic we really like. But can we do a test cross with humans? No, we can't do a test cross with humans. So instead we use something called a family pedigree. And what a family pedigree does is it takes multiple generations of a family and we look at a trait and we identify, during, like grandparents, parents and aunts and uncles, children and cousins, who has the trait and who doesn't. And then we work backwards to try to determine what the genotypes might be. And for an example, when we do this, we let a couple be represented by a line connecting them, and a circle is a female and a square is a male. So this is a couple that had 3 offspring, 1 boy and 2 girls. This couple had 4 offspring, 2 girls and 2 boys, and these two ended up having 2 girls. Okay? Now, if we have a phenotype of interest, we can shade that in. And most of the time they do this with the homozygous recessive condition because it's easier to track genes through the system if all of these shaded individuals are homozygous recessive. Okay? And so to give you an idea what this, how you'd read one of these, all right, so it turns out that attached earlobes are dominant to, or free earlobes are dominant to attached free lobes. Or no, free earlobes are dominant to attached earlobes. Right, so if you're again, circles are females, squares are males. Sometimes if we don't know the sex of a child, we put a triangle. But if you're shaded, we know that you're homozygous recessive, and you've got attached earlobes. Okay, so this female, this girl is homozygous recessive. Both of these males are homozygous recessive. This person is homozygous recessive, and this person is homozygous recessive. Now, let's work backwards. In order for these two to be homozygous recessive, they had to get a recessive allele from each parent. And since each parent does not have attached earlobes, they have to have at least one dominant allele. Both of these individuals are heterozygous. Because this couple had a homozygous recessive daughter, they both had to contribute a recessive allele for attached earlobes. Because this person has free earlobes, they have to have a dominant allele. They have to be heterozygous. Okay. If we go to these two, because this daughter is homozygous recessive and they don't have the attached earlobes, they both have to be heterozygous. Now, for this daughter and this granddaughter, we know they have to have at least one dominant allele. But we don't know if they're heterozygous or homozygous dominant. And so what we do is we put both. It's one or the other. We know it's not this, but it's one or the other. Okay? And that's how you would develop and read a pedigree. All right, so next time we'll finish up Chapter 8 and look at extensions of the law of inheritance.